Good afternoon, everyone. My name is Michelle Oshman. I'm the Vice President of External Relations here at BIO, and we are really excited to be kicking off our August mini semester, a 3-part briefing series all about cell and gene therapy. And I just wanted to thank you all for participating and also thank the wonderful team here at BIO, Dr. Danielle Friend and Barrett Stratford for working so diligently to put this series together, and thank both of our speakers for being part of the conversation today. So with that, I will turn it over to Danielle.
Great, thank you, Michelle, and I'd like to echo what Michelle said. Thank you everyone for joining us today. I'm Danielle Friend, I'm a Senior Director of Science and Regulatory Affairs at BIO. And as Michelle said, this webinar is the first in a series of webinars that we're hosting. So thank you for joining us today. We hope you can join the other webinars that we have planned in the coming future. This first webinar will be focused on really cell and gene therapies and the potential and what those therapies are. So we'll be talking about how these therapies can potentially be curative, and certainly how they'll be targeting diseases that potentially have not had treatments to date. We have 2 excellent speakers for our webinar today, and before I introduce them, I just want to quickly say that our speakers will provide brief presentations. We will then move into a Q&A, which I will lead with a few questions that I've drafted in advance. And in the meantime, as you have questions throughout the webinar, please submit them to the question box. And as soon as I answer or ask the questions that I have drafted, I will turn to the questions that are posed by those of you on the line. With that, I'd like to just ask for, oh, perfect, we have the slide up. I just want to point to the BIO website and let you know that BIO does have a series of resources, including infographics, FAQs, on human genome editing, and BIO position statements. So if you have not taken a look at the BIO website yet to see what we have as far as educational resources around cell and gene therapies, I do encourage you to do so. And with that, I would like to introduce our speakers. Today we'll have Dr. Anju Chawla. She's the Director of Medical Affairs at Bluebird Bio, focused on sickle cell medical lead. And then we also have Dr. Peter Emtage, who is the Senior Vice President for Cell Therapy Research at Kite and Gilead Company. So with that, I will hand it over to Anju to go ahead and get us started.
All right. Thank you so much for having me, and thank you for everyone for your interest in gene therapy and things that it might do. I don't have actually a whole lot of slides today because of technical difficulties, but what I wanted to express a little bit today is to talk a little bit about gene therapy in the sense of what is the patient experience from that, versus all the various different types of gene therapy and what the possibilities are, which my colleague Dr. Emtage will take over and talk about that. So in talking about specifically what is the patient experience and taking that point of view, what does it mean to undergo a gene therapy? It does depend on what type of gene therapy you're thinking of. There's different types for different types of diseases. The one that I will focus on today is a stem cell type of gene therapy, or they call it ex vivo gene therapy, which really means that the stem cell, the blood stem cell, is actually managed and is actually modified. The genes are modified in the lab and not in the person. So with steps like that, what are the things that have to happen? The first thing you need to do is you need to actually get stem cells from the patient. So this does take some work. We used to take it directly from the bone marrow. We would take these stem cells out, modify them in the lab over a period of 6 to 8 weeks, and then have the person back in to reinfuse those stem cells. What we found now is that we can actually stimulate this blood stem cells to come out of the bone marrow and then into the blood themselves, and then actually just take the blood out of an IV, spin out those stem cells, and put the remaining blood back in the IV, back into the person. This process is called peripheral blood stem cell harvest. And with those cells, we take those out and we bring them to the lab. Well, what happens to the person in between? So the person needs to come in. They are going to need 2 IVs. They are going to need a medicine to help stimulate the bone marrow to release the stem cells into the blood. So usually this is a process that does involve an admission and a stay for several days. After that, however, the IVs come out and the patient can go home. When the patient goes home, they generally feel fine. There's usually not much weight in the side effects. And again, they go back to their regular therapy, whatever it is that they've been doing to treat their underlying disease. And then once the cells have been processed, they are called back to the hospital. And when you come back to the hospital for things that use stem cell therapy, you have to get rid of the old stem cells to allow the new stem cells to come in. So when you take out the old stem cells, it's not the surgery, it's not you cut in and you go take out all the bone marrow. It actually is usually done either with radiation or with chemotherapy. And it's usually a high dose chemotherapy where you actually cause all the stem cells within the blood stem cells within the body to be killed. And then you replace them, you infuse the new stem cells in, they move into that nice apartment building of the bone marrow space, and they engraft, so they get into those areas of the bone marrow and they grow and they mature into the blood cells. And the hope is, since they've been modified, as they grow into the blood cells, they make whatever protein, whatever gene that you want to express. In the case of what we've been looking at is actually with sickle cell disease, we actually add in an extra gene into the stem cell. So when it goes into the bone marrow, this extra gene actually makes a repaired or an anti-sickling hemoglobin that works with the sickle cell hemoglobin and prevents it from polymerizing and causing sickle cell disease. So what happens after that? You get infused with the stem cells. You still need to be in the hospital for several weeks because you need that time for that bone marrow to mature and to make its own blood cells so you can get out of the hospital and not be at higher risk for infection. Once this time goes on, what'll happen is that usually there are still appointments that need to happen, usually every few months, to come back to check to make sure the blood's working, to see how the body's reacting, and to see if there's any side effects of the medication, and to see if there's any repair happening from sickle cell disease. Is there anything that's getting better? And generally, in that first year, it's every several months. Once you get a couple years out, it becomes only once a year. Now, long-term follow-up is still very, very important because particularly with gene therapy, all of these therapies are brand new. We don't really know all the possibility of what the long-term side effects could be. So the recommendation is that patients who undergo a stem cell therapy, even if they're not requiring any other medications, even if they're feeling well, that they're followed up at least annually for at least 15 years to check for late effects. So once you're even done with the therapy, you still need to come back. You still need to be evaluated. And we also need to make sure we're updating our patients and our patients update us with what's going on. So for many of our patients, that's kind of the process that goes through. There is some thoughts as far as, you know, what else happens when people lose their disease because they're doing something different. There's also a loss of identity. And so a lot of people wonder, you know, who am I now that I've not have sickle or whatever disease this may be. So they do, there's a lot of thought process that goes into supporting folks as they go through this process and into a new one. So that's really the thought process behind what is the patient experience, which is what my portion of the talk was going to be about. I will leave it up to Dr. Emtash to talk a little bit more about other types of styles of gene therapy, including in vitro gene therapy.
Thank you, Danielle. Is it okay to transition?
Yes, please go ahead.
Excellent. Can you bring up my slides? I think it's from a gene therapy perspective and a cell therapy perspective. I'm just going to give the audience a little bit of flavor for not only what Kite is, but the applications of which we move cell and gene therapy forward, much like Dr. Chawla just did. So next slide, please. So Kite is really a cell therapy company that uses gene modification or gene therapy to ex vivo modify and expand lymphocytes for the application in oncology to anti-tumor responses or driving anti-tumor responses in oncology. So we aim to harness and genetically modify those cells and then put them back in. And I have a slide here that talks a little bit about the vein-to-vein application of what we go through to really generate one of these doses. Next slide, please. So The preference here, the mantra of Kite is really to drive and transform oncology using cell therapy, bringing cell therapy not only to heme, but also to solid tumors and the realm of solid tumors, of having to drive deeper, more durable responses in patients for a long time. So a good example for that is in DLBCL, we've got a response rate at 3 years of around 47%. So this is long-term tangible anti-cancer responses in the relapse refractory setting. But most importantly, it's also critical for Kite to make sure that most of the patients out there that can benefit from this treatment have access, regardless of jurisdiction, cost, or whatnot. So there's really two fronts that we're pushing on consistently to bring patients to a centric position to be able to help them not only from a cancer perspective, but also from an access to the treatment. Next slide, please. So we have two main focus from a therapeutic area. We focus in hematologic malignancies across the board, and we are looking at solid tumor malignancies across the board as well, but the hurdle in solid tumor will require a bit more work as we're dealing with almost a totally different beast. Next slide, please. So our two products that have been approved are Yescarta and recently Tecartus in MCL. Yescarta in DLBCL. And so if you look at the history moving towards CAR Ts, you see that we have the first application of chemotherapy in the 40s, moving into antibodies in the mid to late 90s, and then 2010 on, you had the evolution of checkpoint inhibitors, which really, between the interleukin-2 of the NCI, checkpoint inhibitors, you really started to see the ability to bring the immune system in to drive objective, deep, durable responses in patients. And then in 2017, you had the emergence for the first time of two cell therapies, namely Kymriah and Yescarta, coming into the market in heme malignancies and really revolutionizing how those patients with relapse refractory diseases are treated. Next slide, please. So we focus predominantly at this point with our two commercial products in DLBCL with Yescarta and within MCL with Tecartus. Now, that's the first two commercial programs to be essentially for a company to have two commercial assets in development or in development within life cycle management of broader swath of CD19-based malignancies. Next slide, please. And what I wanted to show the audience here is that you can see that from an ex vivo modified vein-to-vein process by implanting into a patient's T cells using a retroviral vector for delivery, a chimeric antigen receptor, that you can really drive the elimination of kilograms of tumor, large, bulky masses over time will go to such a point that the patient becomes, goes into CR and has a prolonged survival to some extent, you know, as I mentioned earlier, up to 47% at 3 years. So it really is a revolutionary approach to using that patient's own tissue to modify that tissue and redirect it to a cancer. Next slide, please. So you heard me mention the vein-to-vein process. It really is unparalleled in that there's nothing else like it. You see it in gene therapy companies, like Dr. Chawla just talked about how they do stuff. This is how Kite does it. Essentially, a patient comes in, we pull blood, we expand the T cells, we get them activated, we transduce them with a virus that harbors a chimeric antigen receptor. We then freeze and we return back to the patient from a leukapheresis. You get the entire evolution of a cell therapy dose to bring back to that patient. So it's unlike the standard classical small molecule. It's also unlike the standard antibody in that it requires multiple steps that facilitate the generation of an ex vivo modified, gene therapy modified T cell process. Next slide, please. So what's the typical journey here? A patient comes in, they're referred into the authorized treatment center. They come in from the community into the hospitals. They're enrolled. They get blood drawn. The blood is then sent into Kite, usually here in El Segundo, but now with our facility in Europe. The patient undergoes, while the drug is being manufactured, the patient undergoes chemotherapy conditioning, and then Yescarta or Tecartus is given back through a central line within the hospital. And the patient then undergoes monitoring and then will leave the hospital at a point, but has to stay close to the ATC for monitoring in case toxicities pop up. But that really is the typical journey. So being able to take that retrovirus, apply it to that patient's T cells, and really get it back to the individual is part of one of the hallmarks of which the manufacturing tech ops group at Kite has excelled in. I mean, today we can give back greater than 98% of the doses that we are asked to do on behalf of patients, which gives confidence that, you know, from a process, patients will be able to get this life-saving drug. Next slide, please. So we've got a robust pipeline in hematological malignancies. We've got a number of trials ongoing in B-cell malignancies. We're looking at investigating new disease areas like ALL and follicular lymphoma. We also have a number of research strategies that will be translating into the clinic, looking at dual targeting approaches in both B-cell malignancies, multiple myeloma, and we have an AML process and product about to go into the clinic here near term. So we're also, with our parent company, Gilead, looking at combinatorial strategies to be able to facilitate deeper and more durable responses. Next slide, please. So I just wanted to take a little bit to touch on solid tumors. Next slide. Want to allow the audience and you, Danielle, to get to questions. So we've got two approaches. We've got CAR T cells, which is a chimeric, non-natural receptor that we put back into T cells to redirect them to a surface antigen like CD19. On the other side, you have the natural infrastructure of a T cell that recognizes a target of importance, and that's a T cell receptor. It is possible that you can take T cell receptors that are specific, highly specific for tumor antigens, and put those back in mass in a modified cell therapy using a viral approach. So again, cell therapy using gene therapy approaches to modify patients' own T cells. Next slide. We've got two clinical pipeline approaches ongoing, Kite 439, which targets the E7 protein in head and neck and anogenital cancers. And then we have Kite 718, MAGE A3 and A6, which targets cancer testis antigens found in bladder, esophageal, head and neck, lung, and ovarian cancers. Next slide, please. And so the intent here from a solid tumor perspective is, because you are dealing with a subset of patients, we are constantly looking to move the needle in this therapeutic. It's important when you think that of the 620 or so patients that die each year in the US, about 580 die from metastatic epithelial tumors. So this is by far the largest unmet need. And so we're looking at multiple ways of being able to develop these therapeutics and bring them back to patients. Next slide, please. Next slide. So there's a couple other pieces that I think are important here when you think about gene therapy, when you think about cell therapy, and the ability to really disseminate this. One of them is generating a much more simplistic but robust cell therapy process to be able to ex vivo modify and deliver back a more reliable and timely fashion. And this is really looking at automation. There's lots of work ongoing in modifying CD34 positive stem cells. There's lots of work ongoing looking at T cells, dendritic cells, macrophages now, monocytes, and B cells for successful therapy. At Kite, we focused in on how best to contain, automate, reduce the exposure to the external environment, and really create a modular system to be able to bring these back. Next slide. And then I think just as important as we think about how we get these life-saving programs and products out to patients, are starting to think now around not only the modification of a patient's autologous T cells, but also taking, iPSC-derived T cells, induced pluripotent-derived T cells, or NK cells, or looking at healthy donor approaches and being able to really create an off-the-shelf process such that when a physician is requiring a drug to be delivered, they can reach out to the freezer or put a note into Kite to be able to ship to them within 24 hours a product that they can then take into to the treatment of patients. So we believe that this will help get these drugs to patients out in the community. And so this is a big part of our internal process. Next slide, please. So from that perspective, we have internally three real pillars that we think help bring cell therapy, gene therapy, and advance the Kite portfolio. One is pipeline. The other one is business development, knowing that we can't do everything from an organic possibility. We need the inorganic development, as well as academic partnerships and how best to move those forwards using key collaborators, because the science in itself of cell therapy in the oncology space is still very nascent. And so being able to think about how we advance these together across these three buckets is going to be important for Kite and how we do business. So with that, Danielle, I think I'll stop there with the challenge of time and get into some of your questions.
Great. Thank you so much, both Peter and Anju. That was excellent. Maybe before we move into questions, I just want to point one quick thing out to folks that I forgot to mention at the start. We are recording this session, so we will make it available to attendees and other folks after the webinar today. And then I do want to remind everyone to please go ahead and if you have any questions, go ahead and submit them through the question box in the webinar platform. And as soon as we kind of step through some of our initial drafted questions, we're going to plan to turn to the questions that are submitted by folks on the line. So again, thank you, Peter and Anju, for your presentations. I think, one of the confusing things when we talk to people about cell and gene therapies is the terminology that's associated with it. Maybe as just kind of a key take-home point for everyone, could either or both of you comment on what are the key differences between in vivo versus ex vivo gene therapy, and maybe some of the other terminologies that are used to describe those as well?
I can take a stab at that. So, when you think of cell therapy, for instance, or actually let's do gene therapy. So you can have an in vivo gene therapy, which is the delivery of genetic material through a delivery mechanism that can be it can be a virus, it can be liposomal polymers, it can be any number of actual really mechanisms for delivery, including liposomes, dendrimers, cationic polymers, peptides, and whatnot. If you can give that genetic material to a patient, let's say by intratissue administration, IV administration, that would be in vivo gene therapy per se, right? Ex vivo gene therapy is the modification of a cellular product and taking that cellular product to either replace or repair damaged tissue and cells to affect a potential change in the indication or disease that patient is experiencing. And that can be really and truly any number of cellular vehicles and platforms. To date, it's really within the hematopoietic stem cell compartment, CD34 positive stem cell compartment, but it can be skeletal muscle cells, mesenchymal cells, lymphocytes, as for what Kite does, dendritic, and even pancreatic islets. The idea here is to remove cells, make a modification using your preferred method of delivery, be it a virus, be it a nuclease or an enzymatic insertion, and then replace those cells back into an individual. So, Dr. Chawla, I don't know if you add to that.
Sure, just making sure my husband's not a blender on before I speak. So yeah, so I think you've obviously there's a very elegant description. I'm a very simple person, so the way I think about it is that ex vivo or in vitro means in the lab. So the idea of where this genetic modification is occurring. Is it occurring in the person? So as was explained, if you inject the genetic material with some sort of carrier that goes within the body, goes to where it needs to go and produce whatever the gene product needs to be. And then with ex vivo or in vitro means you take stem cells out or you take tissue out, and then you make the genetic modification, and the cells themselves are the delivery mechanism for that, and they are the factories for it. So that's the way I think about it in a very simplistic kind of way.
Perfect. That's great. And maybe a follow-up question to that, again, as we're talking about cell and gene therapies, another terminology that comes up is gene editing, especially in the context of some of these newer technologies. Zinc finger has been around for some time, but a lot of people are talking about CRISPR. Can the two of you comment on kind of what is considered gene editing per se?
Well, geez, gene editing can be, there's a lot of different definitions of gene editing. Again, I'll do the simple explanation, and then I'll let Dr. Emtaj take over because he'll make it much more elegant. So to me, gene editing means anytime you're making a modification in the gene, right? So you're editing the gene. So there's different types of gene editing, and it depends on where you're doing the editing and what gene you're affecting, right? So you can either add a gene, so you can just literally take the gene that functions and stick it in and see if that makes the difference. The other things you can do is you can actually hit the promoter of a gene. So if a gene's making a product that you want, or is supposed to be making something that you want, that you affect its promoter region. So you basically turn on the switch by causing a gene modification there, and it turns on the switch to make your gene turn on and express whatever it's going to express. You can also take a deletion if there's something there that you want to take out. And then something like CRISPR, which is such a cool idea, but it's also it's interesting in how you do it, where you basically direct a pair of scissors to the DNA piece, take that specific piece of DNA out, and at the same time, insert the repaired piece. So that's the kind of the goal of CRISPR. So these are different types of editing, but they're all a little difficult in some ways, and we're just learning the possibilities of what we can do with this. Go ahead. Let's see what Dr. Emtaj says.
No, I don't have. I mean, that was as elegant as it gets. There's nothing else for me to add. I mean, I think modifying by fixing an issue, replacing an entire gene with a knockout strategy. There's really 3 or 4 platforms from an enzymatic perspective that's been used to do that, and those are, zinc fingers, meganucleases, talons, CRISPR, and whatnot. And then there's the older but tried and true mechanisms of lentiviral administration, in some cases retroviral administration. And now what you're seeing quite a bit of is using adeno-associated viruses to mediate the insertion of, a gene into the whole cell's genome, usually somatic cells, and using that as a replacement therapy. So just different mechanisms, but the same underlying pinning, as Dr. Chawla alluded to, is basically the driving rationale for it.
Great, excellent, thank you. So during your presentations today, Anju, you talked about sickle cell, and Peter, you talked a lot about cancers. Can the two of you comment on some of the types of diseases that may be most amenable to gene therapy or cell therapy, and maybe talk about what should we expect to see in the next 5 years or so on the horizon as far as diseases being targeted?
I can tell you that when we think about gene therapy, and this is probably more in Dr. Chawla's area, but I would put quotation marks around the low-hanging fruit are monogenic issues, right? These are genes that enzymatic pathways where there's a known issue, and that if you can causally fix that with a replacement or an edit, then you could potentially bring back the biology and recapitulate and relieve that issue. From an oncology perspective, it's a little bit more of a multifactorial beast, right? Because you're not necessarily just fixing a gene, you're putting a live cell back in, and you're asking it to home to the spot where you need it to, and like the tip of a surgeon's knife, remove that tumor without causing collateral damage. So, you know, being able to think about modifying using synthetic biology and really creating and enabling that cellular vehicle, be it a T, an NK cell, or whatever it is from an oncology perspective, crafting the behavior that not really allows it to compete outside of the tumor. You need something that competes at the level of the tumor cell, but more importantly, within the hostile nature of the tumor microenvironment. So I would say, you know, when you think about the next 5 years for oncology, it's refinement of, you know, bringing cell therapies into earlier lines, like Zuma-7 and whatnot. It's looking at helping T cells and CAR T cells overcome the microenvironment so that we bring this benefit to 100% of patients. And I think that kind of evolves. Solid tumors is a little bit different. We have to ask that solid tumor to traffic to a specific solid mass, get in, expand, and have a biological effect. So when you think about the low-hanging fruit, so to speak, it's going to be refinement on the already existing efficacy and DOR, but then it's going to definitely require crafting the right behaviors to impact solid tumors, which I think is a bit of a different hurdle. And then one last thing here is allo, creating an approach where you prevent graft versus host disease, but you also minimize host versus graft disease so that you get engraftment of a foreign donor's cells with enough time to give you the overarching desired anti-tumor response.
Great.
Yeah.
And Anju, anything to add?
Sure, I've always got some to add. I always like to talk. Which of the diseases that are most likely to get benefit are those where there is a tunneling point, where there's a single point, a single pathway where things have to go through. So in monogenic diseases, such as sickle cell disease, where there's one gene mutation, either you can change the gene mutation, you can shut it off, or you can put something in that competes with it, makes it very clear and makes it well, we thought was an easy target, but it's taken us 30 years to get here, but makes it a lot easier to move forward and to make a difference on that particular gene. But if there's other things, like so with a cancer, if you have a specific cancer with a specific marker that's on top of it, then the idea of being, you can design a gene that reacts, you can stimulate that cell to make the protein to react to that specific cancer cell. So again, where there's a single point of contact, where there's a single pathophysiology, that'll work really well. Where this won't work well is when there's a multi-system, many reasons for a disease to come together. Like, you know, for obesity, for instance, you might have some genetic pathways, but it's not going to be one genetic pathway that affects it. It's going to be 4 or 5 different genetic pathways. So a gene therapy, trying to address one therapy is not going to be enough. You need to address all those different pathways. So I think that's where those will work best, are things that have a single pathway, or at least an area where it's a single pathway before it blossoms off to a lot of different ways. What's going to be coming up soon? Oh, that possibility is, you know, that's like asking for the crystal ball. There's so many things that could be done. Thinking about different types of thalassemia, thinking about different types of cancer, thinking about different types of, like, even juvenile diabetes. If we can figure out that one pathway that's affected, can we use gene therapy to turn on a protein or shut off another one to make it more effective? I think those are the kind of things that we'll be able to do in the near future. Near future, however, again, we thought gene therapy for sickle cell disease was going to be here 25 years ago. So I don't dare speculate. I think we're in a really good place, and we're a lot closer than we've ever been.
And you know, Dan, I'll just add one thing to that. When you think about monogenic, or you think about oncology, part of the ancillary development that has to happen is actually the development of approaches that allow multifactorial, right, edits, right? As Dr. Chawla alluded to, you know, you go beyond the monogenic to maybe 2 or 3 aspects of a pathway, and it's difficult to really conceive of how you can fix that at this point. So there's also a compendial and an associated development in the space of looking at additional ways to multiplex edits efficiently, or kind of deliver entire payloads that reconstitute biological pathways, and that definitely is still kind of evolving at this point.
Great, great. And I think that really segues into my next question pretty well. And I think, Peter, you alluded to this, and I know, Anju, you talked about it earlier in the presentation as well. Some of these technologies are newer, and with that comes some uncertainty. And so I'm wondering if you could talk about some of the uncertainty around cell and gene therapies, and what are we continuing to learn that will, advance the science as we go forward?
So that's an interesting point. I think for that you have to look at the history, right? 1998, 99, we ran into issues with SCID in France, right? The first versions of retroviruses ended up fixing, but also ended up resulting in leukemia. Then, not so much longer after, we had an adeno incident on the East Coast that led to really a setback into how how viral vectors, how gene therapy is kind of done. So I think when you think about the future, and you think about where you bring these various pieces back together, I think when I think about an excitement, when I think about where it will go, to me it really is pushing beyond the hurdles of creating a new vector, creating a new process, optimizing the manufacturing process. It really starts to think about how you bring the bulk of patients in to a response and hold them there for a prolonged period of time. I firmly believe that with the evolving knowledge from an immunological perspective and the ability to really craft the right behaviors, the field of cell and gene therapy is limitless, right? And from an oncology perspective, it is bringing that 80% of cancer deaths a year from metastatic epithelial tumors in. So I think what you'll start to see is a delineation of the pathways that kind of impact patients, the editing, the gene modification, and really endowing the T cell with the ability to compete in that hostile environment and bring benefit. And I take it beyond T cells to NK cells, macrophages, really any cellular vehicle that allows us to do that. And then the last one is essentially the evolution of a universal donor allo system, where we can take these out of a 2-week manufacturing period and really bring them into an off-the-shelf approach to be able to give to patients. So that's kind of what the excitement is. But I do think that we will address a lot of oncology through cell and gene therapy.
And then to take that in the thank you, sorry. And to take that on a non-oncologic side, with really thinking about the kind of advances I'd love to see and what looking forward to is to see if we can find a different way to clear out marrow space without having to use chemotherapy, without having to use radiation. There are some things that are being looked out there using monoclonal antibodies, the bone marrow system, without having side effects that you would normally have with chemotherapy. I think advances in the quality of the stem cell that we are manipulating and bringing in. So how do we harvest? How do we take care of those cells? And how do we do those modifications? And then how we reinfuse? Can we make that even better? Can we make that engraftment better? Can we make it easier on the patient? Those kind of things would be of critical importance, I think, and have to be are being addressed, but we're not there yet. We've finally figured out how to make it work. Now it's a matter of how to make it safer. And we don't know what the long-term effects of any of this is. We really, really need to focus on what are we doing and capturing what can come out of this, because it may not all be bad things that happen, and maybe amazing things that happen as well. So we need to see that, and we won't know it until we do it, try it, watch it, evaluate it. And there's also so many wonderful things that gene therapies can indirectly work with, right? So we talk about it being the thing to help with a monogenic disease, but why couldn't it make tumor cells more susceptible to chemotherapy? Why couldn't it be used to improve the immune system to fight off specific infections? There's different aspects of this that we haven't even thought about yet, or people have thought about but haven't had a chance to bring it forward. So like, I agree. I think the options are limitless, and we're only reined in by how much time we have to try everything.
Great, great. I like that. I like that. I'm going to start now turning to some of the questions put forward by the individuals on the line. So certainly please keep sending those in, but it looks like the first question that came in is around whether currently or in the future you see some sort of combined approach where maybe patients get both a gene therapy and a nuclear medicine, or a gene therapy in addition to a small molecule. I don't know if either of you can comment on whether that's currently occurring or whether we'll see that in the future.
I can take that initially. So I can tell you that there's quite a bit of activity ongoing in combining, a cell therapy from a T cell perspective or an NK cell perspective with systemic, either small molecule or additional biologics. Currently, it's probably the easiest way to assess the combination of multiple mechanisms on the potential outcome with respect to patient responses. As we talked about earlier, it's very hard to contemplate multi-parameter editing to really create those behaviors. So where you have the ability to do a systemic combination to make an assessment from a clinical perspective, it's probably where the field is going to go until we adopt the right technologies that allow us to do that.
Anything from you, Anju, or should I move on to the next question? Okay.
So I think that question was more directed towards an oncologic perspective. So as far as using combination therapy, I don't think we'll be used. So at this point, if the gene therapy is not as effective as we would hope to completely ameliorate whatever disease process it is, then I do think there is a way of thinking about other types of not chemotherapy, but other types of therapies to match with it to optimize one or the other. So I do think that's a possibility in the future, though I think actually in the near future, it's really more on the oncologic side.
Great, thank you so much. So this next question is really around kind of clinical trials in the context of gene therapy. Someone's asking, what do you hear most from patients who are considering participating in a clinical trial for a gene therapy? What are their key questions? What do you feel that you have to educate patients about the most in the context of cell and gene therapies?
I think I can start on that one. So from particularly a stem cell style of gene therapy, the biggest thing is, what are the risks, right? This is something that is irreversible. People do understand that, right? So once you go through this, it's not like you can change your mind. And if you, like with the other medicines that you take every day, if the medicine's not working, you have a side effect, you can stop. Here, once it's done, it's done. So I think that's one thing that's a little more scary that people need to understand, but it's also its advantage. If once it's in and it works, you don't have to take medications anymore. It's done. So again, there's both a positive and negative to that. Right now with stem cell therapy, the idea that this is not just an injection once, this is a process of a period where you are involved, you're still involved with the medical system. And so even though it's a one-time therapy, it's a one-time therapy over time. I think that's one other piece that people ask about. And then the other issue is that the things that come along with using chemotherapy to ablate the bone marrow, the risk of fertility, the risk of late effects that come up. So I think those are all things that you need to take into consideration before thinking about going forward to a gene therapy like ours, like a stem cell gene therapy.
Yeah, excellent. I don't have much to add to that. Kite, if you think about our initial patient populations that were used for registration, I mean, everybody was hopeful, right? That's what we were in the business of, is pulling a patient back from 3 to 6 months of life expectancy and giving them, birthdays and Christmases and Thanksgiving and whatnot with their families. So a lot of what we were asked was, will it work? And I think when you start to see the emerging data, then you realize that, it became, yes, it will. And then secondly, I think the other question that we heard was because it was an immune response that we were eliciting, what's the toxicity, what's the toxic profile and whatnot. And the space evolved in such a manner from an academic perspective into a commercial perspective, because the response rates were so staggering and remarkable in these patient populations. Clinically, we figured out how to manage it, right, to bring it forward. So I think, you know, now it's a good time for those individuals with those types of malignancies, but before it was touch and go because it was new, right? It was still so nascent.
Great. And Peter, I think this next question is geared more towards you, and maybe does touch on some of what you just said. But one question that's coming in is, what do we have to understand around the science of CAR T therapies to better understand any variants that we see in whether a treatment works for one patient, but maybe not as well for another patient with the same type of cancer?
So that's a hugely loaded question, and we can spend like 2 hours on that, but I will try to do my best. So if you think about patient to patient variability, I pull blood, I take an apheresis from an individual, modify it and put it back, right? We are now as a space only really starting to understand what it is within that apheresis that drives the response to CR for some, right? So when you think about it, if I take blood from an individual or from any 10 individuals, the state of those T cells are going to be quite different, right? Patients would have seen different chemos, they would have been open to different regimens, the washout times. So the fitness of those T cells are going to be critical to really crafting and generating that overarching response. So along that value chain, it really is what you take out of an individual and what you put back into an individual, right? I think we've been really good at making sure that patients get their drug product back, you know, be it from our clinical trial design or clinical trial development or commercial process. And so I would say that if you look at where variations can come in, a patient with a pre-existing mutation in CD19, for instance, which is the target of our therapy, will escape. They will go through a response that is not complete. It'll be a partial response, they'll relapse. There are some patients that lose CD19 completely and they relapse. There are others that go on to long-lived, pretty substantial CR rates for quite a long time. And so I think the inherent patient-to-patient variability of their T cells will really kind of determine the potential outcome. And that's where the space of cell therapy is really now starting to understand what those differences are.
Great, thanks, Peter. And this question I think is for both of you. Someone has a question around if a gene therapy is approved by the FDA and that therapy is considered a first in generation, what do we imagine the you know, next generation or the second in generation gene therapies look like? I think the question's asking, would they be changes in the vector that's being used or the method of delivery? I know we've alluded to some of the improvements we might see in cell and gene therapies moving forward, so I'm wondering if you could comment on that.
Wow. Yeah, we need another hour to answer that question. There is so much possibility. So what would be the next generation? Again, I really think now it's going to be, if we've got efficacy, how can we keep that efficacy and improve the side effect profile? I think then also, again, so this idea of will it be a new vector? I think there's plenty of vectors being tested, and I'm sure that there'll be more than one vector type out there for stem cell therapies. I also think that, again, if there is antibody-mediated myeloablation, that's going to be a big advancement, and I think that won't be too far behind. And then really, you know, is there a way to follow these patients more safely, right? Do they have to come in for blood tests? Is there a way? I just was hearing, this is something somewhat unrelated, but I'm just hearing of a patient who has thalassemia has now come up with an app that can guess your hemoglobin by taking a picture of your thumbnail on your phone. So it can determine your hemoglobin level. So there's so many possibilities of advances that can be made that are both directly from gene therapy as well as indirect, that I think we're going to make some amazing progress very quickly.
So it's a really good question. I think for cell therapy, it's it's a little bit probably more straightforward. So as the space has evolved over the last, let's say, 10 years, we've started to understand why patients respond, why they don't respond at all, and why they stop responding. So if you look at any commercialized product from a life cycle management approach, the idea would be, one, do no harm to the current commercial market and build on that to extend the response and safety into deeper, more durable lines of responding patients, but also broader and earlier lines of therapy. So when you start to think about that, an easy example is, about 30%, let's say, of patients getting Yescarta stop responding because they lose CD19. So then the question becomes, can you target CD19 and another B cell marker like 22 or 20 or CD79 to drive the response deeper and capture that population so that you now have a greater level of durability and a higher level of efficacy. So that's a pretty straightforward. If you think about alternatively, not the fitness of everyone's T cells coming in is not going to be the same. So if you go now outside of the life cycle management and you think about changes that you can make within the manufacturing process to bring a much narrower line of consistency between the potential activity of a patient to another patient's come from a comparability perspective, you've got another modification that can come in later, not only in the physical therapeutic, but also within the realm of manufacturing. But if you do change the therapeutic, keep in mind that we're now entering a phase where we're now into life cycle management strategies, and you may be asked to do simple things like randomization against your own initial drug, right? So you have to really sit down and understand the biology that you're modifying and how far or how much of a greater impact to efficacy and DOR you're expecting.
Excellent. Thank you, Peter. And it looks like the last question from our patient groups on the line is around kind of the intersection between maybe science and business. I think the question's trying to get at, what are some of the barriers that you all see in the work that you're doing, and what would you need in order to advance the work that you're doing to overcome those barriers? I know science sometimes just takes time, but I'd be happy to hear your perspective.
Go ahead, Peter, you go first.
All right, so so I think there's phase appropriateness. There is a consistent and a really evolving line of communication as we educate the FDA and the FDA educates us, and it's going back and forth. So I think understanding how to get these therapies into patients in the most safe manner, but also be able to get them in and test, right? So a really quick example of that is solid tumors. If you think about the price of a cell therapy and the overarching output that you need to have in a solid tumor, you have to get pretty close to cure, right? Of that patient. I mean, we're talking an efficacy rate and a DOR that's long enough to support the price of that drug. So being able to work with the agency as to doing phase one, learning, build, and test from a clinical perspective is going to be important. And then I think from a research perspective, we always need targets. We always need the space of immunology to evolve so that we can start looking at pathways that we think would enable our cellular therapies to be much more efficacious. So a number of facets.
Yeah, so exactly that is actually the first thing that came to mind is when we think of these therapies in the non-malignant world where there really is a curative intent. We don't know if they're cures yet, but that's the intent, is to get that level. And what does that mean for somebody who's got a lifelong genetic disease to not have the expression of that disease? So what does that mean? It seems so simple, but really is quite difficult to explain on how do you measure outcomes? How do you explain to the FDA how serious the disease is and what a difference it is to get into a healthy state, not just to a non-sick state? And this idea of, so those are the kind of things that I think of in that circumspect. The other thing is that these things are difficult to do. This is as personalized medicine as you can get. It's for an individual. You're taking their biologic stem cells, you're manipulating those and then putting them back in. How do you do that safely? And then really, how do you scale up so you can get to everybody? And how do you make that process more affordable so it's something that can be done for everybody? So that we're thinking about as far as how do, not only do we've got, I think we're getting to the point where we've got efficacy in some way. How do we improve safety? And then how do we scale up so it's available to everybody? And how do we make that affordable so it's accessible to everybody who has that disease, be it in the US, be it in the EU, or maybe anywhere else in the world?
Great, thank you so much. And we are just coming to the end of our time here. So I want to thank both Anju and Peter for being with us today. I really enjoyed your presentations and the discussion was excellent. So thank you so much. And with that, I will hand it over to Barrett.
Thank you so much, Danielle, and thank you, Peter and Anju, for joining us today. We look forward to seeing everyone next week when we discuss how we will pay for these innovative treatments. If you have any questions in the meantime, please feel free to reach out, and we look forward to seeing you next time.
Excellent. Thank you.
Thank you.
Thanks.
Bye.
Bye.